Hi, my name is Liron Yoffe and I'm from Weill Cornell. I will present our study, single-cell RNA-seq analysis of pre-invasive to invasive lung adenocarcinoma progression. Lung adenocarcinoma is the most common subtype of lung cancer, which accounts for most cancer-related deaths worldwide. The recent widespread use of low-dose chest CT for lung cancer screening purposes has led to the identification of ground glass opacities, GGOs, or non-solid nodules that are not visible on plain chest radiography. These nodules can harbor very early-stage tumors. Growth of the nodules or development of a solid component can indicate more aggressive behavior. To investigate changes in the tumor microenvironment during lung adenocarcinoma evolution, we performed single-cell RNA sequencing of lesions that were resected from 21 patients, as well as matched normal adjacent tissues from the same patients. The lesions were all part solid, meaning they had both non-solid and solid components. Sampling both components from the same patient created a unique and exciting opportunity to observe real changes that occur in each patient as his tumor develops. After sequencing all samples, we integrated the cells and identified 5 main cell lineages, as you can see here in this UMAP. The cells are colored by the major cell lineage. We can see the lymphoid, myeloid, epithelial, endothelial, and stroma cells. The epithelial cells cluster included also the malignant cells. We then characterized those populations in patients' lesions and normal tissue, and today I will focus on describing the immune microenvironment, the lymphoid and myeloid cells. To better examine the changes in the immune microenvironment as the tumor progressed from non-solid to solid, we first pre-clustered the lymphocytes and identified the major cell types, including B, plasma, NK, CD8, CD4, and Tregs. The percentages of B plasma, CD4, and Treg cells were increased in the non-solid components compared to the normal lung tissue. Additionally, NK cells percentages decreased in both the non-solid and solid component of the lesions. Having seen the changes in the cell numbers, we wanted to determine activation status of the cells. We reclustered the CD4 T cells and identified naive, exhausted, and memory effector CD4 T cells. We observed an increase in the non-solid and solid samples of 2 exhausted clusters, first expressing high CXCL13 and the second expressing high TIGIT. TIGIT is a non-immune checkpoint receptor, and CXCL13 is a chemokine produced by human T follicular helper cells. This pattern is consistent with the previous finding that the exhausted CD4 T cells could skew towards the TFH-like phenotype. As mentioned, Tregs were elevated in the lesion compared to normal tissue. We wanted to further assess their naive to suppressive characteristics, hence we reclustered them and identified 5 main distinct clusters. We then calculated a suppressive score and a naive score for each cluster. The suppressive score was based on known signatures that included markers such as CTLA4, TIGIT, and PD1, and you can see the expression of some of those genes over here. Cluster 1 gained the highest suppressive score, while Cluster 0 gained the highest naive score. Additionally, Cluster 0 was characterized by increased expression of TNF receptors 4, 18, and 9, which are known to be highly expressed in tumor-infiltrating Tregs. The IL-2-STAT5 signaling pathway and oxidative phosphorylation pathways that are known to be related to the suppressive activity of Tregs were both upregulated in this cluster. In Cluster 0, the IL-2-STAT5 signaling pathway was downregulated, further suggesting that Cluster 0 represents naive or resting Tregs. Even though there was not a significant change in the distribution of the Treg clusters between tumor and normal samples, both Cluster 1 and Cluster 0 displayed an increased suppressive score in the non-solid and solid components. CD8 T cells did not display significant changes between the tissues. Nevertheless, we reclustered them to find differences in their subpopulations. We identified a very cytotoxic cluster of CD8 T cells expressing GNLY and CX3CR1. This cluster was significantly decreased in early lesions. Interestingly, a previous study by Gao et al. That characterized T cells from non-small cell lung cancer patients demonstrated that CD8 T cells expressing the same markers were reduced in lung tumor samples compared to blood and normal tissue, which was also validated using IF. Our results show that this decline in the cytotoxic CD8 T cells already begins in the very early stage of tumor progression. An additional significant change was an increase in 2 clusters that exhibit high exhaustion scores. One of those clusters also express GZMK, but not other cytotoxic markers such as GZMB or PRF1, which may indicate a transition state of cytotoxic to exhausted cells that are elevated as the tumor progresses. In a similar manner to the lymphoid cells, we reclustered the myeloid cells and identified the major cell types, including macrophages, dendritic, monocytes, neutrophil, basophil mast, as well as the immunosuppressive PMN-MDSC cells. Dendritic cells were enriched in the non-solid samples compared to the normal tissue. We reclustered them and observed a decrease in inflammatory score of CDC2 cells in the solid component. In addition, Langerhans cells that only weakly stimulate T cells were increased in the early lesions. In previous studies, Langerhans cells were shown to be interspread among tumor cells in agreement with our findings. PMN myeloid-derived suppressor cells were enriched in the non-solid samples compared to the matched normal tissue. The inflammatory response pathway was downregulated in those cells in non-solid versus normal, suggesting an additional immunosuppressive activity of those cells. We next reclustered the macrophages and characterized them. While the immunosuppressive or M2-like macrophages increased in non-solid and solid samples, the pro-inflammatory or M1-like macrophages remained the same. Interestingly, the M2 macrophage clusters that express SPP1 was uniquely expressed in the lesions and not in the normal tissue. A recent study identified SPP1 as a potential biomarker for early lymph node metastasis in lung adenocarcinoma. Overall, in the early lesions, there was an increase in total and exhausted CD4 T cells, B, plasma, suppressive Tregs, dendritic cells, PMN-MDSCs, and M2-like macrophages, and a decrease in cytotoxic CD8 T cells and NK cells and the inflammatory activity of CDC2. Those changes in the progression of the tumor suggest that the immune microenvironment changes towards an immunosuppressive and anti-inflammatory pro-tumor microenvironment. Thank you.